Welcome to the podcast called The Battle for African Agriculture, where we're the lens of political economy to look at African food system, African agriculture, and the challenges around it. My name is Belay. I'm the General Coordinator of the Alliance for Food Sovereignty in Africa. I have here with me Professor Dr. Michael Antoniou. Am I pronouncing your name right?
It's near enough. Antonio would be more accurate, but that's good enough.
Anthony.
Not bad as
Anthony.
a first attempt.
Thank you very much for accepting my invitation. Thank you. I thank you on behalf of the African civil society movement. As I told you before we're opening this conversation, a number of, there is a lot of pressure from companies and organizations in Africa to push African countries to adapt GE crops. So they give them a number of reasons for that: productivity, scientific validity, advancement into the future, and whatever. So I'm very, very much happy to explore these issues with you. But for the sake of our guests, can you can you share your background to us? What led you to molecular genetics, and what's your background also?
Yes, thank you, Million. First of all, I want to say that it's an honor to join you on this podcast today and congratulate you and your associates for the sterling work you're doing in raising concerns and challenging the developments of genetic engineering in agriculture, which is very important because, as I hope will unfold during our discussion, your listeners will appreciate that actually what is claimed for this technology and what is the reality are very different. But for me, historically, I'm a molecular biologist. So I got an interest in genetics even as an undergraduate when I was at the University of Oxford, and then went on to do PhD in this field. So so what I ended up specializing in was in the structure and function of genes within a human animal context, though, not an agricultural context. And as part of that, I have used all manner of genetic engineering technologies to investigate the structure and function of genes. And then also, any discoveries that I made, I always look to see if they can be used in a clinical context. So I have
What does it mean? Sorry, what does it mean in a clinical context? You mean in
clinical context? Yes. So what that means is in two areas. One is the development of gene therapy medicines for targeting genetically inherited diseases. And one of my gene therapy medicine designs has been used in clinical trial, which has with a group of patients suffering from beta thalassemia. Beta thalassemia is actually quite similar to sickle cell disease, which is probably be much more familiar to your listeners. The same gene therapy medicine design can be used for both sickle cell disease and beta thalassemia. So that's one area. But another area that I've made major contribution to is in the industrial manufacture of therapeutic proteins such as antibodies. So this is where you use you culture genetically engineered cells, grow them in huge bioreactors, huge tanks, and then you harvest your therapeutic protein from that. And I have made contributions into that area as well in biotechnology.
Congratulations.
So I say this so that your listeners know that I'm not fundamentally against genetic engineering. Obviously, otherwise I wouldn't be doing what I'm doing. But for me, there is a place where you have to draw the line, where you have to also acknowledge the limitations, the technology, acknowledge the unknown dimensions to its application, and draw the line. And where I draw the line is really in the agricultural applications, because whereas when we use genetic engineering in a clinical context, in using genetically engineered viruses and cells and so on in a clinical context, everything is contained.
Mm.
Everything is very strictly regulated.
Mm.
So that the entity, virus
Mm.
cells, cannot reproduce out of control and spread from person to person and cause problems. No, this is not permitted. Whereas in an agricultural context, we have basically the application of very similar, if not the same technology, but where organisms are being genetically engineered and then put out into the environment, which for me, not fully acknowledging the imprecision and unpredictability of the technology. And as a result, with their release into the agriculture and by default into the wider environment, they possess an innate and inherent, unknown, unpredictable health consequence for the consumer, whether they be animal or human, and also for the wider environment, their impact on the wider environment. So for me, this is, as I say, where I draw the line. And I put that on from a scientific evidence-based perspective. It's not a theoretical one we're talking about here that I'm objecting, raising concerns about. It's actually what the fundamental science of genetics and the limitations of the technology tells us how we should be very cautious about its application in an agricultural context, if at all, if we are to use it at all.
Yeah, I mean, most of the time, their argument is we wear clothes which is coming from BT cotton, chocolates where one or another kind of sugar added from BT grown materials. We have for our breakfast different kind of things. They can be genetically manipulated. So we are actually eating it. Nothing has happened to us. So, that's what they say. That's an argument.
Yes, indeed. I hear these arguments all the time. I've been sorry, I've been hearing these arguments for decades, actually. But the point is that there have been no controlled epidemiological surveys of human populations that are consuming genetically modified foods to show whether there have been adverse outcomes or not. So you cannot make this claim. Yes, maybe perhaps people are not dropping dead, as it were. But there could be. The point is that if they're causing things on adverse ill effects, it may take time, could be long term, and if those problems can be conditions that are perhaps fairly common already, like allergies, food intolerances, then it may be difficult to spot. So there's no epidemiological evidence of no harm, but where we do have strong evidence, some good evidence, and I would say quite strong evidence of potential harm stemming from the consumption of genetically engineered, genetically modified foods, is from controlled laboratory animal feeding studies.
Yeah.
Here, where you can control things. Yes, you have the animals eating the GM corn, the soy, or whatever, and then to animals that are on standard diet. And under these controlled environments, you can pick up on any adverse outcomes. And for me, there are a sufficient number of well executed studies that show that the consumption of GM corn, GM soy can lead to quite a wide range of adverse health outcomes, but particularly in terms of liver and kidney function, but also digestive system problems and immune system problems. These are documented. This is documented evidence under controlled laboratory conditions, which cannot be denied. And for me, these animal studies, because they're using animals that are standard surrogates for human health, assessment. We have to take them seriously. And we should be made aware that the consumption of these same foods by the human population could be causing adverse outcome. And let's face it, there are, illnesses that used to be very uncommon are now very common. Allergies, food intolerance, digestive system problems like inflammatory bowel disease or cardiovascular problems, metabolic problems. These diseases, and even more serious things like even cancers. These are diseases that used to be very uncommon 60, 70 years ago. And since then, these have become very common in populations around the world. So we have to ask ourselves the question, why? What has
changed?
What has changed? And of course, many things have changed, Micheal, without a doubt. There are many things. But the one thing that has changed, which affects us all, is the way we grow and consume our food.
Yeah, definitely.
And that includes the introduction of genetically modified crops. And we have to bear in mind that the introduction of genetically modified crops comes with new exposures that can lead in the longer term, if not the short term, to adverse health outcomes. There are, just to put things into a broad perspective, there are three possible sources of a possible health risk, adverse health outcomes that can arise from the consumption of GM foods. One is the product of the foreign gene, as in genetically modified crops, at least the old style gene addition, what we call transgenic crops, which is probably at the moment what would be grown in Africa and most other territories, is you have a foreign gene from another organism. So, for example, with the BT, the GM corn, which has BT toxin, there's the gene for BT toxin is from a bacterium. So this gene from bacterium has been put into the corn to make this BT toxin that is supposed to act as an insecticide. Now, this may act as a reasonably effective, and I would say reasonably, it's not 100% effective against the target pest, but it could be toxic to the consumer.
Yeah, it's the same.
This BT toxin has never been an integral part of the human diet. So what it may have an effect. We do know, for example, the BT toxin is highly immunogenic. It can cause a strong immune reaction in the consumer. And so the question that arises is, could that lead to allergies? Could that lead to gut problems? The BT toxin could be compromising our gut integrity, like it does, for example, in the target pests. These things are possibilities that have remained under-researched, underexplored, and remain possible sources of why people have reported allergic reactions from, for example, from the consumption of BT toxin genetically modified corn. So that's one possible source of adverse reaction is the actual foreign gene product itself. The other is the pesticides that are still used with the cultivation of GM crops. And particularly here, and this has been an area of my own research of my own group over the years, for over 10 years now, my group has become, I like to say, a world leader in the researching the toxicology of glyphosate herbicides. The glyphosate GM crops tolerant to glyphosate herbicides were launched back in 1996 and have spread in numerous areas around the world, in various countries around the world. What that means is that people are exposed to much higher levels of glyphosate herbicides now than they were before the introduction of genetically modified crops. So this is another source of health risk, the higher residues of pesticides, particularly glyphosate. And last, and not least, and again, this is an area of particular interest to me, given my background in molecular genetics, is that the genetic engineering process is not precise, in that it brings about a wide range of unintended DNA damage, what we call mutations.
Mm.
And this wide range of unintended DNA damage can bring about the disruption of many genes. When you disrupt the global patterns of gene expression of a plant, of a crop, it could be corn or whatever, the plant may look fine. But you see it grows, produces the corn, fine. But its biochemistry may have been markedly altered and part of that altered biochemistry from the changes in gene function caused by the unintended DNA damage or mutations is that this could give rise to toxins and allergens, unexpected production of new toxins and new allergens. Nobody is looking at this. Again, although we do have animal studies that point the finger that the GM engineering, genetic modification, genetic engineering process has disrupted gene function with unknown health consequences. Certainly, there's no way you can call the GM and the non-GM somehow substantially equivalent and therefore not requiring any health risk and no monitoring. This goes contrary against what the science tells us. Work in my own laboratory, for example, compared a variety of glyphosate-tolerant GM corn with equivalent non-GM corn. And when we looked at the composition of the two, we found major changes in both the protein profile and the biochemical, small molecule biochemical profile. And we can definitely say that the reason for this difference was caused by the genetic engineering process. The disruption to the genetics are caused by the genetic engineering process. And others have found similar results. So all this claim that the genetic engineering is actually a very precise procedure is not underpinned, not supported by the science that underpins this technology.
Yeah, that was excellent. The three areas of concern. But there are some who say that, because of CRISPR, which led to gene editing. They say that, the previous generation of GMOs are not that much precise, but now we got the medicine, there's no need even for mixing to unrelated organisms. We can actually do gene editing and it's very much precise. And whatever is produced out of that process is very healthy and very progressive. What would you say to that myth?
I'm glad you called it a myth, Micheal, because that's exactly what it is. It is a myth. Because again, the science does not support these claims. The advocates of gene editing in an agricultural context. Again, we have the same gene editing technology being applied clinically in a what I would consider as a very cautious, very sensible way, with careful safety assessment for unintended genetic changes in addition to the intended change. And of course, the patients are monitored very carefully. But again, we see the same technology applied in an agricultural context, and the advocates are arguing for total deregulation.
That's what they're pushing for.
It's staggering. It's absolutely staggering to me how you can be, in a way, so dishonest to the science that underlies this technology. Because again, what can happen is this with gene editing. With gene editing, as you were hinting, what it does is rather than introduce a foreign gene, you're changing one or more genes that are already there in the plant or the animal, because farm animals are also being gene edited as well. Let's focus on the plants, because for me, that is where the main worry lies, with the gene edited plants. So you change one or two genes in the crop, whatever it may be. And then you say, oh, well, I've only just changed these genes, and maybe this could be these changes could even happen naturally. Therefore, I'm only mimicking what may happen in nature, and therefore, why should this be regulated?
Yeah, that's what they say, yeah.
This is what's been advocated for deregulation. It ignores two major facets of the technology. These claims ignore, conveniently, two major facets of this technology. Firstly, is that no gene works in isolation. All our, whether it's us as humans or any other organism, plant or animal or human, all genes are working as part of a network. If you change just one gene function in a network, you're going to have effects on many other gene functions and biochemical functions. So it's not, even in terms of outcome, it's not a clean out process. You may say, I changed this gene, and this gene normally, the protein from this gene has this effect, and that's all that's going to happen. No, that view is too narrow, because that function is part of a network, and it's going to have biochemical consequences way beyond your intended change. So that's one thing that's not acknowledged. The second is that the gene editing process as a whole is not that different from the old style transgenic genetic engineering process. What you do is you take your plant cells, you grow them in the laboratory on dishes, what's called plant tissue culture, and then you introduce your gene editing agent into the plant cells, and then you allow it to do its business, and then you grow new plants from the plant cells. Now, this whole process, the plant tissue culture and the gene editing transformation process, these are inherently highly mutagenic again. There are hundreds, if not thousands, of unintended sites of DNA damage in the genome, in the DNA of the plant cells growing on the dish. So when you grow the new plant that has, which you screen for the gene editing event you want, at the same time, you're going to be carrying forward, like I say, hundreds, if not thousands, of sites of unintended DNA damage along with it. So the same applies here as with the old style transgenics. This wide-scale DNA damage is in some of that DNA damage may not have any consequences on gene function, but many will. And so you can have alteration, DNA damage in genes, disruptions in gene functions that can lead to altered biochemistry, that can lead to novel toxin and allergen production. This is a reality. This is not fantasy again. This is a known outcome of the process of the technology. And this is what is being ignored by those that are advocating for deregulation. And what trick do they play to try and convince the regulators of this? They say, just consider the final product and not the process of gene editing.
Yeah, that's what they are.
Because they know that if you consider the process as a whole, then you would have to acknowledge the wide-scale unintended DNA damage and its consequences that you would need to look for. And it's possible to look for it using in-depth, what we call molecular profiling methods. You would have to, if you acknowledge the process as a source of damage, then you would have to regulate it, and you'd have to ask the developer to look for it. But because the developers know this, which is why they try to detract. They never, if you look at all of their materials, or if you ever get into discussions with them, they will never ever talk about the whole process of gene editing. They will only talk about final products.
Yeah, that's definitely the case. Well, so you've touched it here and there, but in a very concise way, what are the off-target impacts of gene edited crops?
Yeah. So there are, like I mentioned, the off-target outcomes of gene editing are twofold again. One is off-target damage by the gene editing tool, the CRISPR.
Yeah.
The CRISPR can modify the part of the DNA that you want, the on-target site, although there can be problems there too that I'll come to in just a minute. So one is the CRISPR can damage the DNA unintentionally at various locations around the gene. One. But also in addition to that, like I mentioned before, just generically, just the growing of the plant cells, the plant tissue culture, growing of the plant cells in the laboratory, and the transformation process, the introduction of the gene editing tool into the cells, this has even wider scale DNA damage, which is also off target. So combined, the CRISPR off target plus the tissue culture transformation process off target combined can amount, like I say, to hundreds or thousands of sites of unintended DNA damage. And that's why it's so worrying, because invariably amongst all of this DNA damage, you're going to affect the functioning of multiple gene functions. Like I mentioned to you earlier in the one study from my own group where we compared the GM maize with the non-GM maize, we found many changes in composition, which could only arise from changes in gene structure and function. There's no kind of other way. So that points to that even in other words, in the final marketed product, because we did the molecular analysis in a marketed GM maize variety. So in the final marketed product, we could have compositional changes, unintended compositional changes, the GM or gene editing transformation process as a whole that have gone completely uninvestigated and unregulated and with unknown health consequences for the consumer as well as the wider environment.
Excellent. Let's explore issues around productivity and food security. What they claim is through genetic engineering, they do two things. One is increased productivity, yeah, and the second is reduce the amount of pesticides that are used, especially the BT cotton, BT maize, and all the BTs. So to begin with, is food security a question of availability or productivity?
Yeah.
That's one is a very important question. And will the GE technology address food security?
This very good question again, Micheal. This is a fundamental question because, as you know, the justification of this genetic engineering technology, including gene editing, is that we need it to feed the world, to more food security in the face of climate change and all of this sort of thing. Now, however, when you explore the subject carefully, it's very clear that actually the world, we produce too much food already anyway. In data available, this is data that is several years old now, and things have not changed, I'm sure. But in research sponsored even by the World Bank, as well as the UN Food and Agriculture Organization, concluded actually that we produce enough food now to feed 14 billion people. In other words, almost twice the population we have now. So needing more food now to feed the world's expanding population is a myth. The problem of why people go hungry now is not from a lack of food, but from access to the plenty of food that is there already. So in other words, it's poverty that needs to be addressed. In other words, it's a socioeconomic problem, not a productivity problem. So making these arguments of needing genetic engineering, gene editing of crops and animals to help us in this way, to save losses and so on, is to make more food available is just a myth, really. It's just for me, it's more like emotional blackmail to try to convince people that you need it. So that's one thing. But let's look at what the technology can deliver
So you were responding brilliantly, about, hunger is not an issue of production alone.
Mm.
And you're saying that there's a lot of food, according to the data from World Bank and others, there's enough food in the world.
Mm.
One of the question is access, because
Mm-hmm.
people are poor, even if food is around them, they cannot access it. And also the food might not be useful to them,
Mm-hmm.
So that can be a problem. But if you go back to GM crops, what is the data of their performance up to now? Are they actually
Mm.
much more productive than conventionally produced
Mm.
crops,
Yeah.
Yeah, just that
The
question.
very good question again, because again, higher productivity, higher yields was one of the claims and
Yeah.
justifications for the introduction of GM crops. But actually, there has not been any increases in inherent yield from any GM crop or gene edited crop for that matter up to now. So the claims of high yields have not been met at all. And in some early research work that was done in the United States, where they compared productivity of GM soy against non-GM soy, they actually showed that the yields from the GM were lower per hectare than from the non-GM. So, and that again, I did not find that surprising because, if the genetic engineering process disrupts gene functions, it can reduce the performance. One of the outcomes of disrupted gene function and biochemistry is that the crop will perform less well than better. And again, we have to remember one thing about characteristics or traits such as yield are not just down to one gene or two genes.
Yeah.
Yield is like a whole physiological response of the plant. It's how you grow it, and its total, and the functioning of the totality of its genetic makeup, not just down to one gene or another. It's what we call a complex trait.
Yeah.
A complex genetic trait is where you have many gene families working together as a network that contribute to the performance of the plant. Other complex traits can be disease resistance or pest resistance. These, again, are not just down to one gene, but due to many gene families.
And the drought resistance
And
too.
drought resistance, all of these highly
That's why they
desirable.
couldn't produce
Exactly.
any crop which is drought resistant, because it's a combination of so many,
So many genes. Absolutely
yeah.
right. So that's why what we find is that genetic engineering, either old style transgenic and also the new gene editing, for me, in terms of giving you these complex traits such as higher yield, drought tolerance, heat stress tolerance, pest resistance, or pathogen resistance, high yields, are doomed to fail. They're just not going to work. It's the science, the molecular genetics, as we understand it now, genome biology, totality of gene makeup biology, tells us that you're not going to succeed through manipulating one or two genes or introducing one or two genes. You are not going to successfully, in a robust way, enhance these complex traits.
Yep.
Simple as that. For example,
yeah.
just to give you, say, for
Yes.
example, you change one gene to make the plant resistant to a pest attack or an infection from a fungus, a pathogen attack. How long will it take for that? And it's just one gene or at most two genes. How long will it take for the pathogen to adapt to that one gene change before it bypasses your engineering and now infects just as bad as before? I would say we know these organisms change incredibly fast. probably one growing season, two growing season, and you'll be back to where you were before. It's I'm afraid, as I say, it's just not. And this is why where we have had major successes in enhancing yields, enhancing drought tolerance, enhancing resistance to pathogen infection and pest resistance is through natural breeding, where we take two varieties of corn or whatever plant we like, and we naturally mate them and then select for the right gene combinations. Why? Because now we're selecting for the multiple gene families that are going to confer the complex trait for us. And when we combine these together like this, it will be, first of all, effective, and it will be robust. So, and there has been many through this process of genetic marker assisted breeding. There have been many successes in selecting for example, pathogen resistant millet in Africa. Higher pathogen resistant millet has been developed using this type of procedure. And that's just one example that I'm aware of. And probably there are many others as well that are out there that are now benefiting farmers and people.
You know, some countries grow GE in a large scale. The US is one of them, and Brazil, and India. What kind of lesson would African governments take from the experience of these countries?
Yeah, that's a good question again. There are multiple lessons for me that can be learned from this. Firstly, that these crops offer no advantages over conventional crops. That's the first thing. They will not increase yields, and they also will not reduce pesticide inputs. Take, for example, the glyphosate tolerant, herbicide tolerant crops. You're still having to use large amounts of herbicides, pesticides in combination with these crops. And because the weeds have become, and actually things have got worse, because the weeds have become resistant to the glyphosate. So now farmers are having to use combinations of herbicides. The industry has launched crops that are tolerant to multiple herbicides and not just one herbicide. So the chemical inputs have actually escalated over the years from the introduction of GM crops rather than reduce. And that also applies to BT crops as well. BT was supposed to replace spraying. BT, you know, the engineered GM crops, BT crops, were supposed to reduce pesticide spraying. But actually, again, first of all, it was only a partial protection in the early days. It was only partial protection. So the farmers still had to spray some. And then, of course, as the insects develop resistance, they have to spray higher amounts of insecticide and combinations of insecticides to control. So no enhanced performance, no reduction in chemical inputs are lessons to be learned by Africa from the GM farming in North and South America in particular. The other thing that Africa needs to be, that African farmers need to be wary of is the fact that the industry, these GM seeds are patented. They're owned by the biotechnology industry, like Monsanto or Syngenta and BASF, you know, these, or Corteva, you know, these major companies. They have exclusive patent rights on this. So what that means, they have full control of the seed. So firstly, the cost of these seeds is much higher. The farmer cannot save the seed to plant the next year. They have to buy the seed new every year. And so the costs for the farmer to grow GM crops is much higher than growing conventional crops, which are just as good anyway. You know, it's not as if there's any advantage to be gained. They're just as good, if not better, than the GM anyway. So, and so what farmers in North America and South America are finding, for example, is they're trapped now with these contracts with the industry that it controls. The industry is controlling what the farmer does and doesn't do through the patent rights that they have over the seeds. So African farmers need to consider this as well. Do they really want to be like a slave to the biotechnology industry who is controlling what they can and can't do with their seeds?
Yeah.
And also, and with the higher upfront costs as well. So I think these are two very important lessons that Africa should learn from especially from farmers in North America, and think very, very carefully about whether they want to buy into this technology or not, because there are major consequences for them economically, without a doubt.
Thank you very much. Let's go to an area which is very, very much alive in our context, and that you were an active participant to, which is the Mexico report that you've written.
Yes.
I'm an Ethiopian and we love eating injera. Injera is made of teff. Teff is a native crop to Ethiopia. It's endemic to Ethiopia. And, teff is cultural, teff is our identity. I've been told that, I think I read somewhere that over 6,000 accessions are in the gene bank of Ethiopia. It's a very, very critical crop in every Ethiopian. If I'm sure, I hope you went to one of Ethiopian restaurants, and if you have eaten injera, you know what I'm talking about. So now they have gene edited teff, and it's quite a shock to us all. But I think what I'm going to ask you about Mexico is related to this, because corn or maize is close to the heart of Mexico. It's their identity in it. It has originated from there. It's everything to them. Economy, social, cultural, and everything. So I just wanted to bring one of your recent contribution that feels relevant for Africa right now. You prepared a report for the Mexican government as it was moving to restrict GMO corn imports for health reasons. In that report, you cited a large body of evidence from well-controlled laboratory animal studies showing harm to multiple physiological systems from genetically modified corn. Now, this is striking because Mexico took that evidence seriously enough to justify national restrictions, even under strong international pressure. In Africa, we are facing similar pressures to adapt or import GMO. So I would like to ask, what did you find in that research, and what lessons do you think African governments can draw from Mexico's example?
Yeah. So, yes, it was an honor for me to contribute this report to the Mexican government. And indeed, they took it seriously because the science is good. It's solid evidence that there can be health risks from the consumption of BT, especially BT GM corn, and especially the newer varieties there. We've highlighted how in the first early generations of BT corn, maybe there was one or at most two different BT genes in the crop. Now you have 6 of them, and they're combined with herbicide tolerance as well. So you're having exposures to multiple BT toxins and multiple herbicide residues, insecticide residues as well on top of that as well. So you have to take on board the fact that the animal feeding studies that showed evidence of harm from the consumption of BT corn were with the early generation BT corn, with just one or two BT toxins. Is there going to be? There's a possibility that could be amplified further, of course, with the newer generation variety with many BT toxins, and as well as heightened in pesticide residues that automatically come with that as well. But even with these early studies, I kind of alluded to the kinds of outcomes earlier, where there were even the studies conducted by industry, by Monsanto, submitted as part of their to the regulators for market approval. Even the industry studies showed that there were multiple statistically significant biochemical changes in the animals fed the BT and glyphosate tolerant GM corn, suggestive of kidney and liver disruption, kidney liver functional disruption. This is very clear, and there were follow-up, in addition, as follow-up studies by people in academia seem to confirm that as well. And this comes earlier, as I say, that these negative health effects can be coming from the consumption of the BT toxin, or from biochemical changes, from the disruption, from DNA damage in the crop. You know, the studies conducted, you can't distinguish between these possibilities. Perhaps it's a combination, but there's the finger clearly points to the genetic modification, whether it's because of the BT or the genetic damage induced by chemical changes. In a way, is irrelevant. It's the point. It's the GM feed that is the problem. So this is, and the same applies. Now you mentioned the teff, which, by the way, I have eaten and it's very tasty. And it's very sad for me to hear that this has been genetically engineered. But can you tell me what has been the genetic engineering that has been done with the teff? Can you give me a bit more information?
Yes, they say that you know, when it rains, the soil will be waterlogged.
Yes.
And when that happens, if the plant is tall, a single plant, then it bends. You know, it will lose all the seeds. So what they are claiming is they are producing a waterlog resistant variety, where it is very short and it wouldn't bend that much in the breaking. That's what they say.
Okay. And this is again, is it gene editing? CRISPR
gene editing. Yeah.
CRISPR gene editing.
Gene edited, yeah.
Yeah. So they must have disrupted one or two gene functions that make a short teff plant rather than a normal tall teff plant. This is what it sounds like. Which again, you look at it at face value, you think, yeah, that's great, isn't it? Imagine all I've done is make a tall plant to a short plant, or surely there can be no harm in this.
Yeah.
But as we discussed earlier, what is being ignored in the claims that this is no issue to be concerned here is the fact that these gene functions that have been changed, what other gene functions that they're connected with have been altered? What off-target DNA damage has been caused by the CRISPR, and what off-target DNA damage has been caused by the transformation process, the tissue culture and the transformation process, which, as we discussed before, will amount to hundreds, if not thousands, of sites of unintended DNA damage. What are the consequences of this? So yes, the teff plant may grow and produce maybe good seeds, but what about the composition of those seeds? What is the biochemical changes? And I can almost guarantee you that if they look carefully, they would find changes. They will find changes if they did the analysis properly, because the technology exists for us to do what is known as molecular profiling. This is where you can look at the total spectrum of proteins and the total spectrum of biochemicals, small molecule biochemicals. And this is what you need to use in order to find out the consequences of your gene editing. It's not just a matter of saying, oh, yes, you see, I've disrupted this gene and this gene in the way that I want, and so everything is fine. No, show me the
the bigger picture. Show me the big biochemical consequences by doing what we call metabolomics, biochemical profiling, and proteomics, protein profiling. Show me this as to, because only this tells you what is the total effects of the gene editing process. Not that you've changed one gene, because you know there will be other genes disrupted and there will be consequences to this. And so this is what I hope whoever the regulators are in Africa that are dealing with this submission, they must embrace the total science that underpins the gene editing. They must embrace the process as well as look at the end product and ask for proper risk assessment based on process as well as end product. Only then will they get a total picture of what is going on. Exactly. And this must include not just biochemical analysis, protein analysis, but it must include toxicity studies in animals as a result as well. Otherwise, you are just assuming and praying. You know, you put it out and pray that nothing goes wrong. Not on the basis of science, but based on almost faith. And that's not what it belongs here.
Thank you, Micheal. Our time is running. We are about to wrap up.
Okay.
An important question is, I think you've responded to me about climate resilience, you know. They say that they will produce, or they are producing drought-resistant crops, you know.
Yeah. The thing, this is a good point, Micheal. I'll mention this just very briefly. The point is that there's nothing wrong with the crops and animals we have now. What is the problem is with our system. We need climate-ready, climate-tolerant systems of agriculture, not climate-ready crops or animals. Our animals and our plants, absolutely fine. They've been bred by people for many years and they're very sturdy. We now need to change our system of agriculture to an agroecological, a diverse agroecological farming practice that will give us the resilience and the security in our food system.
Yeah, exactly. What about, I mean, you explained really, really well the health and also the climate-related issues. And the politics of it. What about the ethical issues and impacts on environment?
Yes, indeed. There are ethical ramifications, not least of which, as I mentioned earlier, is that these gene-edited, genetically modified crops and animals, they're not aimed at the small producer. Anyway, they're not aimed at the small-scale farmer that dominates world agriculture, more than 70%, and much of it in Africa that way as well. They're aimed at the big producer. And so they're really not going to help the small-scale farmer that dominates agriculture anyway. They really aren't. And as I mentioned earlier, it really just, because of the patent control, the control based on the patents that the industry has over these seeds, these developments, they exercise for me inordinate, unethical control over the farming system of what farmers can and can't grow. So I think this is also this should be a concern as well of any nation that is considering adopting these crops on any scale, and look for alternatives. Yes, I mean, I've alluded to already on this, but the point is, I have not to this day come across any genetic engineering, gene edited application that has been offered that you cannot achieve it better through an already existing system.
Very good point.
Really haven't.
It's a very, very good point. Yeah. Let's go to politics of GM in Africa. Why do you think is Africa targeted now as a new frontier
Now,
of
I should
GM? Would say that.
yeah, again, very important issue, but I'm afraid I'm not up to speed on the politics of this in Africa, I'm afraid. So I can't really give you a clear answer to that. But obviously, Africa is seen, generally, the industry sees Africa as a major market for their products, of their biotechnology, genetically modified, gene edited products. And so they are clearly trying to work their way somehow. Again, you will know more about this than myself in terms of how they're trying to work their way into the political system and influence governments in Africa. I think what I've seen over the years and what other commentators have mentioned is how obviously African countries are strapped for cash. And so the big biotechnology industry here, like Bayer, Monsanto, and Corteva, and Syngenta, and so on, it's been evident that they're using their financial might to buy their way in to the systems in Africa, where they can sell their products and expand their profits there too. Not that Africa needs their products. In fact, the evidence of especially crops that have been developed specifically for Africa, such as the virus-resistant cassava, was a complete failure. Genetically engineered virus-resistant cassava was a complete failure. Not surprisingly for me, again, when you look at what was attempted to be done there. Whereas, of course, you can
the beta-carotene fiasco,
the beta-carotene fiasco as well. I mean, these, as if you needed, a specific plant to be higher in beta-carotene, where you can grow beta-carotene rich crops in a diverse agricultural context without any trouble at all. So I think looking historically, it's been a failure. Genetic engineering attempts in Africa have been a failure. And as I mentioned earlier, I think by looking at the systems and supporting farmers to put in better systems of agriculture, supporting them for better agroecological, diverse farming practices, this is where they will gain security and they will gain independence. There will not be under the control of foreign influences. They will be their own masters and be in the control of their own production and not be in the pockets of foreign companies. So
thank you so much.
That's as much as I can say.
No, thank you so much, Michael. Let's end in a positive note.
Yes, of course.
Are you hopeful? And in what kind of agriculture do you see hope?
I see hope in people like you. I hear many good things of NGOs, campaigners like yourself, working together for a better agriculture in Africa that is free from the shackles. And that's what it is, free from the shackles of genetically engineered crops. So that gives me great hope. And I wish you all the best in spreading the truth about these things and the sensible alternatives. And I have seen interviews of farmers in Africa who have put in place the agroecological practices on their farms. And they are so inspiring, where they tell you before they were struggling, and now with the help, with the right support, they put in the right agricultural practices, agroecology, mixing their bananas with the cassava, with all these other diverse crops. And now they have food security. They're making money because they're good yields and able to sell. And that inspires me as well. So we need to get the messages of these farmers that have succeeded with the alternatives out there as well. and for the as I say, for the small farmer, which is the vast majority in Africa, as well as globally, to adopt, and that is for me the way forward for the African continent.
Thank you very much, and that's what we are pursuing as the Alliance for Food Sovereignty in Africa.
Beautiful.
I hope we'll bring you to some African countries, and you visit, the farms that you.
I would be an honor. It would be.
Described.
I would really love to visit some of these places because they're so inspiring.
Yeah, definitely.
And the people are just wonderful people.
Yeah,
you can feel such warm-hearted people are really connected with nature.
Yeah,
this is what you.
Antony, been in England and I visited a number of farmers. I talked to them, and I've been in the US. I've talked to farmers everywhere. They're nice people, personally.
They are.
Yeah, because they're close to the soil.
Exactly.
The land is very, very much ground to earth. So
yes,
they're all nice people, and it's very sad to see this happening to them. You see.
Indeed, I agree with you.
Yeah,
but there is, as I say, there is hope. The precedent is there. The hope is there that we will move in this positive direction in the future.
Yeah, we have to strive together to get there.
Absolutely.
Thank you so much. Thank you.
You're very welcome. Thank you very much.
Really
it's been an honor.
Thank you. Thank you.
Bye.
Cheers. Bye.
Bye.